Dear viewers, greetings. In this video, we are going to see about colony counter. The contents of this video contains an introduction about colony counter, principle of colony counter, types of colony counter, instrumentation of colony counter, steps for using a colony counter, applications of colony counter, advantages of colony counter, limitations of colony counter, and precautions for using a colony counter. Colony counter. A colony counter is a laboratory instrument used to accurately count the number of microbial colonies, either bacteria or fungi, growing on a petri dish or similar surface. A colony counter is a device used in microbiology laboratories to count bacterial or microorganisms colonies which are grown on a solid growth medium. It simplifies and accelerates the colony counting process, providing an accurate measurement of colony forming units or CFUs in a given sample. Colony counting is essential for numerous microbiological applications, including drug development, food safety testing, and environmental monitoring. Colony counter system streamlined this process, allowing researchers to obtain precise colony counts in a fraction of time it would take using manual methods. Various types of colony counters are available to help enumerate colonies of bacteria and fungi quickly and accurately. Some of these colony counters are manual, while others are automatic. Principle of colony counter. The basic principle of a colony counter is to detect and count an individual colonies on the surface of an agar plate. Colonies represents the growth of microorganisms that arise from a single cell, and their numbers can be used to estimate the concentration of microorganisms in the original sample. Modern colony counters use digital imaging technology to detect colonies based on their size, shape, and color. The machine processes these images to differentiate individual colonies from the background and other artifacts. Types of colony counter. There are 4 types of colony counter. They are manual colony counter, semi-automatic colony counter, automatic colony counter, and digital colony counter. The first type of colony counter is manual colony counter. The manual colony counters are simple devices equipped with a light source and a magnifying glass to help the operator manually to count the colonies. The second type is semi-automatic colony counters. The semi-automatic colony counters involve some manual work but provide electronic counting aids such as clicking a pen-like stylus to mark the colonies, and the counter increments automatically. The third type is automatic colony counters. The automatic colony counters use cameras and image processing software to count colonies automatically. They are fast, efficient, and reduce human errors. The fourth type is digital colony counters. The digital colony counters are high precision machines that scan plates using sensors or cameras and digitally count the colonies using software algorithms. Instrumentation of colony counter. The colony counter contains 7 major parts. They are illumination system, magnifying glass or lens, pen stylus or clicker, display unit, camera, computer or processor, and control panel. Illumination system. An illumination system provides even lighting, making the colonies visible for counting. It can include a backlit plate holder or ring light. Magnifying glass or lens. The magnifying glass or lens magnifies the colonies to aid in manual counting. Pen stylus or clicker. This pen stylus is mainly used in semi-automatic models. This pen stylus is used to manually mark the colonies, and the machine counts each marks. Display unit. The display unit shows the number of colonies counted. Camera. The camera is present only in automatic models. The camera captures images of the plate which are then analyzed by software. Computer or processor. The computer or processor processes the image data and calculates the number of colonies. Finally, control panel. The control panel allows the users to input settings and control the machine's function. Steps for using a colony counter. There are 6 steps. The first step is sample preparation. Prepare and inoculate an agar plate with the sample. The second step is incubation. Incubate the plate to allow microbial colonies to grow. The third step is plate placement. Once colonies have grown, place the plate in the colony counter. The fourth step is image capture, particularly for automatic colony counters. For automatic counters, capture the image and let the software analyze the plate. The fifth step is manual counting, if using manual or semi-automatic. For manual models, count the colonies by clicking or marking them. Finally, result display. View the total count on the display panel or software output. Applications of colony counter. In microbiological research, colony counters are used in laboratories to estimate microbial load and contamination levels in research samples. In pharmaceutical industry, colony counters ensure the sterility of drugs and pharmaceutical products by counting bacterial colonies in sterility tests. In food industry, colony counters assess microbial contamination levels in food products to ensure food safety. In environmental monitoring, the colony counter is used to assess the presence of microorganisms in water, soil, and air samples. In medical diagnostics, the colony counters counts bacterial or fungal colonies in clinical samples like urine, blood, or wound swabs for diagnostic purposes. In public health, colony counters are used in microbial quality control of drinking water, dairy, and other consumables. Advantages of colony counter. The advantages of colony counter includes time efficient and suitable for high throughput operations, highly accurate and reduces human counting errors, user friendly with automated counting functions, reduces eye strain and operator fatigue. Data storage and export capabilities enhance the traceability. Differentiates colony types for detailed analysis. Standardizes counting for consistent result across users. And finally, customizable for a range of microbial assays and plate types. Limitations of colony counter. The limitations of colony counter includes difficulty in detecting small, faint or overlapping colonies, limited capability for morphological analysis, requires regular calibration and maintenance for accuracy, compatibility issues with non-standard plates or colored media, high initial cost and limiting accessibility for small labs. Lighting and background color sensitivity affecting detection accuracy, inflexibility for custom applications or mixed microbial populations, risk for over-reliance leading to oversight, requires training to operate advanced features, struggles with plates showing high colony size or colony size variability. Precautions for using colony counter. The colony counter should be placed in a flattened, dry place. Add dust cover on colony counter after use. The colony counter should be protected from moisture, direct sunlight, acid and alkali erosion. Prevent bacterial culture from contaminating the counting pole. When counting the colonies, the probe pen should not be tilted too much and the number will be entered when you tap it gently until there is a sense of bouncing. The colony counter and the probe pen should not be disassembled at will. Finally, use purified water to clean magnifying glass and other surfaces and then wipe it with lens paper before use. Dear viewers, that's all about the colony counter. Thank you for the support. Thank you.